Today, we're going to talk about Western blotting. Western blotting is used to identify specific proteins from a complex mixture of proteins extracted from cells. The first step in a Western blot is to homogenize your tissue of interest. Place the tissue in a tube full of beads. This tube is then shaken at high speeds to break down the tissue and make something that looks a bit like a cell milkshake. This new slurry is then removed from the beaded tube and is placed in a microcentrifuge tube. This tube is filled with a buffer that contains urea and some amount of SDS page. The SDS page covers the proteins in your mixture to ensure they are separated by size and not charge. Immediately after this step, you can quantify how much protein you have by using a nanodrop machine.
So now that you have extracted your protein samples, just put it aside. And now we'll move on to the next step, which is preparing the gel. You're gonna prepare the 10% gel electrophoresis like we usually do in lab. And then put in the comb and let it solidify. Meanwhile, you're gonna pour the running buffer into the electrophoresis tank. Then, after the gel has solidified, take the comb out and put it into the gel tank. Then load the gel with the ladder and the protein samples. Run your gel until the samples reaches the bottom of the gel. And now we're going to do the electrotransfer process. In this process, you're going to make a gel sandwich. You will have 3 filter papers soaked in transfer buffer shown here in green. Then you're going to put your gel and SDS page together shown here in red. Then you'll add another 3 filter papers also soaked in transfer buffer. And finally, you put a sponge shown here in yellow. After you made your gel sandwich, you're going to put it into the transfer apparatus and run it at 4 degrees Celsius for about an hour. After the transfer process is done, take the SDS page out.
Now this is the SDS page out from the gel sandwich. The next step is the blocking stage. Here in this slide, it is showing a 2-sample run, and since the SDS page membrane will have the protein all separated by their size, it will look like the slide you see right here, except you wouldn't be able to see these with the naked eyes until you image it. And let's say this is the protein that we are interested in. Here is what it looks like from the side view. The yellow circle will be the protein that we are interested in detecting. For the blocking stage, you would add the blocking membrane with the 5% skim milk in TBST for 1 hour. And to detect our protein that we're interested in, we will add our primary antibody in the bovine serum albumin, and you will simply incubate this overnight at 4 Celsius degree on a shaker. So this is what would be going on if we were able to visualize the protein and the antibodies on the SDS page. Then you wash the membrane with the TBST for 5 minutes for 3 times to get rid of all the antibodies that are bound to non-specific protein. After removing the unbound primary antibody, the membrane is exposed to another antibody. These secondary antibodies are usually bound to biotin or other reporter enzymes such as horseradish peroxidase, which will bind to one primary antibody and enhance the signal so it could be detected when you image it. Next, you will take the SDS page into Western blotting imaging machine, and there are many different types out there, just like we have the 2 types in our class. So this is what you will see after you image it, that our antibody is bound to our protein of our interest, and there it is.